The overall goal of the following experiment is to genotype multi-drug-resistant Mycobacterium tuberculosis using an amplification microarray. This is achieved by performing a multiplex asymmetric PCR over the top of a microarray within a single microfluidic reaction chamber. There, the sample is simultaneously amplified, labeled, size selected, made single-stranded, and hybridized to the microarray. Next, after the microarray is washed, a reader shows if Mycobacterium tuberculosis is present and if it contains a DNA mutation known to confer resistance to rifampin, isoniazid, ethambutol, or streptomycin.
The main advantage of this method over existing microarray, line probe, or nucleic acid hybridization methods is that an amplification microarray significantly simplifies the user interaction with the test, still while providing the analytical sensitivity of PCR and the multiplexing capacity of a microarray.
For this procedure, have a clean workspace wiped down with decontaminant solution and have nucleic acids pure enough for multiplex PCR. After thawing and mixing all the stock solutions, assemble the PCR master mix. When preparing this mix, make at least 1 extra volume of mixture. For the TB test shown here, also add extra Taq polymerase to the master mix in addition to the amount of Taq provided with the stock reagents. Mix 1 microliter aliquots of sample DNA to 49 microliters of master mix. Carefully add 48 microliters of each.